Mhm. Thank you, Dr. Gray, for the introduction. So, yeah, as Dr. Gray mentioned, my name is Eman, and today I'm going to talk about a part of my PhD project. My PhD project is focusing on investigating the tumor microenvironment in malignant pleural mesothelioma. So I'm going to start with saying that malignant pleural mesothelioma is a malignant tumor that arises from the pleural lining of the lung. And actually, a couple of weeks ago, Julia talked about the pathogenesis of malignant pleural mesothelioma. So I'm not gonna go through that again today. However, I would like to highlight that malignant pleural mesothelioma has the second highest epithelial mesenchymal signature of all cancers after sarcoma. So if we look here, we see that malignant pleural mesothelioma is classified histologically into three subtypes, which are epithelioid, biphasic, and sarcomatoid. Actually, epithelioid is the most common. And going back to the idea of the EMT, I think this also supports the presence of three histological subtypes, moving from epithelioid into sarcomatoid and malignant pleural mesothelioma. And then I think most of us know the story about anti-PD-1 and anti-PD-L1. So anti-PD-1 and anti-PD-L1 improve the prognosis of cancer, of many types of cancers. And actually, this is also the case for malignant pleural mesothelioma. However, there was a clinical trial, which is Checkmate 743 clinical trial that has been done in a group of mesothelioma patients. And they compared immune checkpoint blockade, specifically anti-PD-1 and anti-CTLA-4 versus chemotherapy in malignant pleural mesothelioma patients. And if we see the Kaplan-Meier plot here comparing the survival of the immune checkpoint molecule group in blue and the chemotherapy group in red, we can see that the survival is much better in the group that received immune checkpoint molecule compared to the chemotherapy group. However, if we look at the median survival, we still see that it's terrible, it's around 18 months, the median survival for this group of patients, even with anti-PD-1. And this is why much more research is needed in this aggressive tumor. So lots of immune checkpoint molecules are actually identified in the tumor microenvironment. So normally, immune checkpoint molecules could be either inhibitory or could be stimulatory. And normally, also again, there is a balance between the stimulatory or the inhibitory effect of the tumor, of the immune checkpoint molecules when they bind to their ligands. However, what happens in the tumor microenvironment is that cancer actually stimulates more or express the inhibitory immune checkpoint molecules more, and this would lead to the immune suppressive microenvironment. And this actually explains why anti-PD-1 alone might not be effective in establishing a very good prognosis in cancer, and that's because the tumor microenvironment is very complex, and blocking one pathway alone might not be effective. So we hypothesized that the quantitative measurements of tumor-infiltrating immune cells and immune checkpoint molecules will identify distinct tumor microenvironment phenotypes that could predict the response to different therapy approaches in malignant pleural mesothelioma. And for my project, actually, I have three aims. The first aim is to identify tumor microenvironment features predicting resistance or sensitivity to anti-PD-1 with or without chemotherapy. The second aim of my project is to quantify the expression of targetable molecules in malignant pleural mesothelioma. And then the third aim is to determine if the detection of epithelial mesenchyma transition at molecular level improve the prediction of treatment response better than histological stratification. Today, I'm going to focus on the second aim of my project. So in order to achieve the second aim of the project, we are going to perform multiplex immunofluorescence using the AstroPath platform. And I'm going to use this panel in order to identify the targetable molecules that could be present in malignant pleural mesothelioma. So we've been optimizing this panel, and this panel is actually composed of 7 targetable molecules in addition to DAPI, which is a nuclear staining. The targetable molecules are CD155, TIGIT, B7H3, VISTA, TIM3, TGF-beta, and FOX2. And as a reminder about the AstroPath platform that Michael presented last week, is that what we do is that we acquire whole slides rather than selecting high-power fields, and then we do image processing in order to generate our data in the end. So why did we end up selecting these specific markers? We have been going through literature in order to see what are the markers that are being targeted now in early phases clinical trial, and also what are the markers that could be expressed in malignant pleural mesothelioma. And that's why we landed on these markers in specific. Actually, a couple of weeks ago, again, like Julia went through VISTA in details, and I'm not going to go through that today. And I have to acknowledge that Trisha spent some time explaining to me Hasta la Vista Baby Joe. I didn't know anything about that. But anyways, I'm not going to talk about that today. And I'm going to focus on three molecules today, which are CD155, TIGIT, TGF-beta, and FOX2. So let's start with TIGIT and CD155. So TIGIT is expressed on most of the immune cells, especially tumor-infiltrating lymphocytes. And actually, the ligands for TIGIT are CD155, CD115, and CD112. However, TIGIT binds mainly to CD155. So this is the main ligands for TIGIT. So when TIGIT was knocked out, it was in natural killer cells. It was found that the cytotoxicity of natural killer cells increased. And the number of clinical trials using TIGIT in different types of cancers are around 70 clinical trials, and 9 of them only are phase 3 clinical trials. The rest of them are early phases clinical trials. So expression in malignant pleural mesothelioma, it has been found that TIGIT is expressed in malignant pleural mesothelioma by doing single IHCs. And then let's move to CD155. So CD155 is expressed on tumor cells, expressed on B cells, as well as dendritic cells. And we can see here that also CD155 ligand is TIGIT, so both of them bind to each other. Another two ligands for CD155 is CD96, which is also inhibitory, in addition to CD226, which is a stimulatory. However, when TIGIT and CD155 bind to each other, this would block the stimulatory effect of the CD226. And when CD155 was knocked out in mouse models, it was found that the pro-apoptotic signals increased and also cell cycle arrest took place. The number of trials for CD155 now is only 12 in different types of cancers, mainly glioblastoma. That's what I found. And the expression in malignant pleural mesothelioma has not been studied. So we're very interested in seeing that in malignant pleural mesothelioma. So let's look at the molecular level, what happens when TIGIT and CD155 bind to each other. So the first cell that we are always excited to see what happens with is CD8 T cells. So when the binding happens between tumor cells and CD8 T cells, recruitment of SHIP1 and SHIP2 takes place, and this will lead to inactivation of CD8 T cells. Another important cell in the tumor microenvironment is regulatory T cells. So if we look here that when the binding takes place between TIGIT and CD155 on tumor cells and regulatory T cells, this will lead to stabilization of FOXP3, and this will lead to the activation of regulatory T cells, which is immune suppressive cell, in addition to the production of TGF-beta and IL-10. And then when we look at the natural killer cell, when the binding occurs between tumor cells and natural killer cell, what will happen is that SHIP1 will be recruited, which will inactivate mitogen-activated protein kinase, and this will lead to inhibition of natural killer cells. Also, when we look at the binding between natural killer cells and dendritic cells, or the binding between B cells and dendritic cells, this will lead to the inhibition of the release of inflammatory cytokines such as IL-12, IL-17, and chemokine receptor 7. And all of these factors together will lead to an immune suppressive microenvironment. So this is actually just an example of the expression of CD155 and TIGIT when we were optimizing these markers in our lab. So this is CD155 in a case of tonsil. And as we mentioned, CD155 is expressed on B cells, dendritic cells. And as we can see here, this is most probably a German center, and these are most probably, again, like B cells expressed in tonsils. And maybe these are, I don't know if these are dendritic cells or not. And then if we look at TIGIT, also again, we were optimizing TIGIT in single IHC and single IF in tonsil again. And this is the signal that we were giving. And this is a multiplex image of both of them in lung adenocarcinoma, CD155 here in orange and TIGIT in red. So we can see here in the tumor, CD155 is highly expressed and TIGIT is expressed in the immune cells in the stroma in between the tumor. So now I'm going to talk about COX-2 and briefly about TGF data. So let's start with COX-2. What is COX-2? COX-2 is an enzyme that produces prostaglandins, and prostaglandins are actually inflammatory. So what are the COX-2 inhibitors? Non-steroidal anti-inflammatory drugs or aspirin. And it has been shown that aspirin, like when you start taking aspirin at an early age, it has been now associated with a decreased risk of developing colorectal cancer. And this is actually attributed to the fact that COX-2 is related with the creation of an inflammatory tumor microenvironment. So let's look at the cells that express COX-2. COX-2 is expressed on most of the immune cells as well as the cancer cells. When COX-2 was knocked out, inhibition of tumor progression took place. The number of clinical trials of COX-2 inhibitors now in clinical trials is around 300 in cancers. And this is not only treatment, this is either like they're testing prevention or treatment. And the expression of COX-2 in malignant pleural mesothelioma using single IHC was found also to be positive. So here, if we look at COX-2, which is produced from tumor cells, COX-2 will lead to production of prostaglandin E2, and prostaglandin E2 will inhibit the cytotoxic activity of CD8 T cells and will cause activation of regulatory T cells. And then COX-2 itself will lead to the recruitment of M2 macrophages, which in return will lead to production of COX-2. And actually, I've been reading like in literature that COX-2 might be responsible for polarization of also of M2 macrophages, but nothing is sure yet. And then on the cancer-associated fibroblasts, again, it recruits cancer-associated fibroblasts, which will produce COX-2 again. And then on the tumor-associated neutrophils, COX-2 actually stimulate the production of elastase in tumor-associated neutrophil, which will lead to the progression of tumor. So this is an example of COX-2 expression in breast cancer. Again, while optimizing it, we used cancer tissue in order to optimize COX-2 since it is highly expressed in cancer. And this is a single IHC, and this is how it looked like in single IF. So COX-2 also plays another role in the whole tumor progression rather than only initiating like an immune suppressive microenvironment. So COX-2, as we mentioned, is responsible for the production of prostaglandin E2, which is again an inflammatory. Prostaglandin E2 upregulates different pathways that would lead to tumor progression and metastasis, including apoptosis resistance, angiogenesis, metastasis, epithelial mesenchymal transition, cancer stem cell stability, inflammation, and tumor growth. And since we're talking about mesothelioma, actually epithelial mesenchymal transition is very important to us. Other than prostaglandin E2, COX-2 itself also plays a role in upregulating HIF1 alpha, which is responsible for angiogenesis. It also upregulates TGF beta, which is one of the markers that we have on the panel, and it inhibits E-cadherin. And both of them will be responsible for the induction of epithelial mesenchymal transition. And also it upregulates microRNA 665, which will be responsible for tumor growth. So as we mentioned now that COX-2 is responsible for the upregulation of TGF-beta, and actually both of them are responsible for the induction of epithelial mesenchymal transition. COX-2 is responsible for that, whether through production of prostaglandin E2 or through the inhibition of E-cadherin or microRNA 665. Another thing that TGF-beta is responsible for EMT mainly through the induction of transcription factors, which are snail, ZEP1, and twist. And also it inhibits like the production, the E-cadherin, which will lead to epithelial mesenchymal transition. And that's why these two are very important for us since malignant pleural mesothelioma has the second highest epithelial mesenchymal transition. This is a multiplex for TGF-beta and COX-2 together in lung adenocarcinoma. And as we can see here, this is the tumor cell. And we see here that both of them, like whether TGF-beta or COX-2 are expressed on the tumor cells, and also they are expressed in the immune cells in the stroma between the tumor. And we would be very interested to see if COX-2 and TGF-beta would be expressed in sarcomatoid or biphasic malignant pleural mesothelioma, since they are related with the epithelial mesenchymal transition. So the cohort that we are going to use for this study is the N227 clinical trial. This clinical trial recruited 520 mesothelioma patients in Canada, France, and Italy. And they randomized the patient into a group of patients who received chemotherapy, another group of patients who received chemotherapy and anti-PD-1 or anti-PD-1 alone. We will have access to around 200 to 250 slides or pre-treatment tumor specimens where we can test this panel. So I spent the last year or maybe more optimizing this panel. So from the beginning, like we selected this markers, and then we did optimization of the panel. So what are the steps that I did for optimizing this panels? I started with single IHC optimization. So single IHC optimization is important because this is our reference or this is our control. We use the single IHC expression of the markers in order to go back at each step of IF optimization to make sure that it's matching. So in single IHC optimization, I was like trying different primary antibody dilutions and incubation times, along with different types of epitope retrievals, and also different time of epitope retrieval and protein lock. And the number of the slides that we used for optimizing the 7 antibodies was around 350 slides. And this is because actually we use around 5 cases for each step to make sure that the expression is the same and we're not like, biased by anything. And then we moved to single IF optimization. In single IF optimization, I was trying to choose like a good secondary antibody, and then I did primary antibody dilution, and also I played with incubation times, and then the TSA fluorophore dilution to get the best signal. And for this step, we used around 455 slides for all the markers. And then in the end is the validation. What is the validation is that we combine all the protocols that we optimized in single IF optimization, and then we do like a multiplex protocol, and then we compare the signal that we're getting in multiplexing with the single IHC to make sure that it's working when combined together. And the number of the slides for validation is 32 slides, only if we're lucky, only if it worked from the first time. And we're not sure if we're lucky or not, so yeah. I've just completed the validation last week, the staining test. And this is how the panel looked like in lung adenocarcinoma. So this is the multiplex, and this is the signal for each marker when I turned off the other markers. And these are the IHCs that we are going to compare with. And for this step, what we do is that we use an image analysis software to count the number of positive pixels, both in multiplex IF and single IHC. So we would be able to see if there is a difference between both or not. And if we're getting like a huge difference between both, this means that the marker needs to be, there are some stuff that needs to be tweaked. So yeah, this is what I'm gonna do like probably this week. And then what are the significance and future directions of this project? So first of all, we need to identify the cell lineages involved in the expression of targetable molecules. So we mentioned that the targetable molecules are expressed in different types of cells like CD8 T cells and stuff like that. So the next step is that we have to identify which cells are expressing that by adding a number of markers on the targetable panel to identify that. And then we believe that the first aim of the project will provide a broad map of the PD1, PDL1 axis across malignant pleural mesothelioma. And then also the second aim, which is about the targetable panel, will provide expression maps of new potential therapeutic targets informing the design of future clinical trials. And also from the data that we are going to get from the targetable molecules panel, we can identify specific molecules that could be added to another panels to identify different features. And then in the end, I'd like to thank everyone in the Kuchan lab. I'd like to thank everyone here for listening and every like collaborator with us. So yeah, thank you everyone for listening.
Thank you very much.
Thank you.
Questions? Right, yeah.
It's awesome. And very curious. I didn't know the CD1 to CD5 and sorry. How would that affect? Because we only had one antibody in between. That are you going to get too much TIGIT? Yeah, TIGIT.
We have TIGIT in CD155.
Yeah, but are you going to get the TIGIT blocking or touching to receptors of CD155?
Oh yeah, that's a great question. So we are like, as Michael mentioned last week, we talked about the spatial relationship, right? So I think we would be able like to study the neighbors of these markers. So I think we can identify like different cells that are close to each other based on the expression of the markers. So I think we'd be able like to know, you know, like the tumor cells are close, for example, to natural killer cells or CD8 T cells based on the spatial relationship data.
I'm so confused though, like the TIGIT could bind to the CD.
Block each other, you mean?
Yeah.
They are not co-expressed. So TIGIT is expressed on T cells, and then CD155 is expressed on the tumor cells, dendritic cells, and B cells. So they are not co-expressed on the same cells.
Oh, I guess I misunderstood the slide. I thought they had the same ligand.
They bind to each other, but TIGIT is expressed on mainly the T cells, and then CD155 on the tumor cells mainly. So that's why when they bind to each other, inhibition of the tumor microenvironment, like the immune suppression takes place.
I guess it's conceivable, depending on where the epitopes of the antibody are mapping, that those epitopes on each of the proteins might be close enough when they're engaged.
Yeah,
Cause a
yeah,
problem.
yeah.
Angie?
Great talk, really cool project. Are there other panels that already can identify things like cancer-associated fibroblasts? And just wondering if that can be related to your signal for your TGF data.
Yeah, we actually, like, there is a panel being developed now related to the cancer-associated fibroblasts. We don't have it on the project yet, but I think this would be very cool to have because I think it's gonna tell us a lot about the EMT, and it would be related for sure with TGF-beta and COX-2.
Yeah, because the other thing, like TGF beta can be made as a latent factor, right? And so maybe looking downstream of some of these could be helpful in the future. I know it's a lot just to get this far, but something like a thrombosbondin 1 is quite helpful to show cells where that TGF-beta pathway is firing as opposed to the cells that are producing it. If you
Yeah.
follow me.
Yeah, I agree. I think that would be very interesting to do. Like, yeah. Yes.
That was a very, very good thought. Just a technical or logistical question. Do you, how many antibodies can you put on at the same time and then do you strip them and repro with other antibodies? Like, how do you just decide which of those two options to do and what gets you to sort of the size of the big pen? How big a pen can you get to? Sure, you can come in something. Give me some guidelines for people.
Yeah, so I think that's an interesting question. So what we're doing now is that we're doing 9 markers. We've been doing 6 before. For the targeted panel, I think it was very challenging to propose the sequence itself and to propose the pairing of each antibody to the TSA fluorophore because most of them are co-expressed. So most of them are expressed on the same cells. So I think this was the challenge. And that's why we spent like long time trying to optimize it. And even like when we reached the validation, we were like, I was so stressed, I was so nervous to see the results. So, I don't think it will be like feasible to do for now, to do more than 9, like especially working with a panel like that. And that's why for adding the other markers for the lineage cells, we're thinking of doing not TSA-based multiplex. We're thinking of doing the DNA barcoded multiplex to avoid like blocking retros and stuff like that. So I hope this answered the question.
Yeah, I think that it's interesting and daunting to think about because even the order that you do them, they
Yeah,
change the outcome, but I guess the codex approach, everything goes in all at once then, right? Or do they do batches with the DNA barcoded antibodies?
we're not gonna do the codex, we're gonna do the UltiView. So the UltiView, they do 4 and then you wash and then you do 4.
4 antibodies at a time.
Yeah,
Okay.
and we were thinking actually of doing 4 only. So CD8, CD163 for the macrophages and cytokeratin for the tumor. So the main ones.
So the
and-
tissues are fixed and robust enough to survive multiple iterations, so.
Yeah, because it's only 1-day protocol, and then we take the cover slip off and we use like a specific mountain for that, so we can take them off. It's not dry, and then we can add the TSA-based technology.
Other questions? If not, join me in thanking Eman for a wonderful talk.